KEET TV presents Homework Hotline, the after-school show that fuses learning with fun. Watch local teachers bring the classroom on air and online. This is Homework Hotline.
Hi, welcome to Homework Hotline. My name is Dan Scofield, and I teach science at St. Bernard's Academy.
And my name is Rita McConnaughy, and I'm a math specialist with Northern United Humboldt Charter School. And you're watching Homework Hotline. Thank you for joining us.
Yeah, welcome to Homework Hotline. Yes, we're so glad you can join us today. Today, we are going to examine that crazy molecule called DNA.
No way.
Which stands for deoxyribonucleic acid. Big long name. So we usually refer to it as just DNA. And oftentimes we'll see it coiled up like this. We'll see models of it, and we'll see a spiral of it, and then somebody will go, hey, that's DNA. And so, you know, what makes DNA look like? We're going to get some DNA. And
it looks pretty colorful and fun.
Yeah, it is colorful and crazy and fun.
Are they this big?
They can be, actually a single DNA molecule is actually longer than that, but not that big.
Okay.
But it can be. It can be super long. If you stretched it all out, it's crazy long because it's so thin. Mm-hmm. Anyway, usually you can't see DNA. Typically it's transparent. I mean, you can't see DNA unless you do special things with the cell. DNA you can see it. The time we do see it is when it's during cell division, we can see it because it gets all packed up in chromosomes, and we have a slide to show you what kind of that would look like, what those chromosomes would look like. And this is a karyotype of the human DNA and human chromosomes. So we have 23 pairs of chromosomes. You get one chromosome from one parent, and you get one chromosome from the other parent. And so we're called diploid for that reason, because the D means two, diploid means two, so we get one from each.
So each of the 23, one is from one.
One's from mom, one's from dad.
Okay.
And so, and they're all, there's 22 what are called autosomes. They look the same on both sides, and they're numbered from biggest to smallest. So the number 22 chromosome is like the smallest one. And then there's one pair of what's called sex chromosomes, which are the X and the Y, or the X and X if for female. And so that's when you see them. And actually what you're seeing there with the chromosomes isn't just all DNA. It's actually a mixture of DNA and protein called chromatin. It's not just pure DNA, but they condense, and that's when you can see the DNA, because it's all packed together and because it needs to get all separated out and divided evenly amongst the cells. So when cell division takes place, every cell gets the same stuff, and so they can make exact copies of each other. And so the cell is all organized in that way to go and divide it up.
So the X-looking things are not just DNA, but protein and...
Yeah, protein is called chromatin together. Okay. But it's a mixture of DNA and protein. In fact, when people were first studying the secret of life and what is it that has all the instructions to make you the way you are and me the way I am and carry out all our processes, a lot of people originally thought it must be proteins, because there's 20 different types of amino acids that make up proteins. And they knew about DNA, and they knew that DNA was in chromosomes, but they didn't think there was enough information in it to go and carry out all those instructions. There's only 4 different kinds of nucleotides that make up DNA, and we'll talk a little bit more about that. But so they thought there was probably it'd be more likely to have it's like a 4-letter alphabet to make all the stories of life, where proteins are made up of like a 20-letter alphabet, and that was what was controlling all the meaning and everything behind it. But so lots of it. We'll talk a little bit more about the studies that went on to figure that out. But let's. Are you going to talk about like the year and everything? Like
maybe
some. I mean, it was a lot of this took. Yeah, we could talk about some of the year stuff that happened.
Like 19.
It was the 1940s when it started in the early 1900s. Was when they started figuring out what DNA. What makes the instructions for life was taking place, and then there were several experiments that went on. But really, it was in the mid 1940s when they discovered that it looked like DNA was all the instructions were on the DNA, and then it was the big quest happened after that was like, how was it doing what it did? How was it instructing how is DNA instructing living things to go and make what they were supposed to make and control the cell in that way, and all living things? And that was the big secret of it. And mainly was the structure. What is the structure of DNA? They didn't know that it was coiled like this, and it took several researchers to do that. And but if you wanted to have a good read about it, here, I'll show you this. This is a really great book. It came out last year called The Secret of Life, and by Howard Markel. And it's a really great read. Lots of information, lots of great intrigue in it. And so a lot of people.
So like if you're in high school, you can read that.
Absolutely,
Middle
high school. Middle school probably too, if you wanted to. And in adults, high school, middle school, it's a great read. Anyway.
Maybe elementary, it would be.
Probably a little too much for elementary.
So maybe read 7th grade on up.
Yes,
6th, 7th
I would
grade
say
on up.
those grades on up. So what I want to do today is let's take a look at some DNA. Let's extract some DNA.
We're going to get to look at DNA? That's
yeah, we're going to look
amazing.
at DNA.
Do you need like a microscope?
Well, we can see it. We can have the DNA, and we'll be able to look at it and extract it from. I'm going to take it out of strawberries, because you know what I said, that we are diploid, meaning that we have 2 copies of DNA. And
is this a diploid DNA?
That's a single strand of DNA.
Just a single one, and if there was. I'm just
2 of each chromosome. So it means that we have all the strands are all packed up and bundled up with proteins and wound around the proteins and all those other things to make up the chromosomes.
Okay.
And so we each have 1 copy from one parent and 1 copy from another parent. So we're diploid. Plants can get kind of polyploid sometimes. They have multiple. Usually, if you have an extra chromosome or 1 short of a chromosome, people don't typically survive that way. There are some kind of cases where they can, but usually too many chromosomes or not enough chromosomes is not a good thing.
I connect that poly, because we use poly, that prefix in math quite a bit.
Many, but some plants seem to get along okay by having extra chromosomes. Bananas are triploid. They have 3 sets of chromosomes. And strawberries are octoploid. They have 8.
Octoploid.
Yeah, they have.
Wow.
2 sets of 4 chromosomes, or 2 sets of 4 chromosomes. So they have
2 sets of 4.
But they only have 7 chromosomes. Like we have 23 pairs. They have 7 groups of 8. So
is there anything else that has 23 chromosomes? Or are we like?
I don't.
Not that you
think. Not that I know, but they could.
So human being most unique.
Well, we are everyone. Every single thing is unique. In fact, some have more than 23. So we don't have the most.
Okay.
So, and these have more chromosomes than us. They have 56. We have
56 chromosomes.
They have 7 times 8, right? 56. We have 2 times 23.
So we have 46.
Yeah. So since this has so much DNA in it, we're going to use it.
I should write that math out. Okay, so you're giving
I'm going to give you a bag.
We're
going to each do this because that way maybe it'll work once.
Okay. So does it matter, like the leaves, would they have different type of DNA?
Actually, that's a great question. Every single cell in the plant will have the same DNA.
Even in the leaves?
Every single cell in us has the same DNA.
Wow.
If it has DNA in it, it has the same DNA.
But they look so different.
I know. And it makes it.
So if this leaf got separated from this particular strawberry, could I match it back up with its DNA?
If you had the technology to do it, you probably could.
All right. Just wondering.
No, that's a great question. All right. So let's take a look. Let's see. Remember we said DNAs were in cells?
Okay.
DNA. So let's take a look at this. We have a cell picture just to remind everybody what a diagram of a cell looks like. All these different parts. This shows a plant cell. And you see that on the outside there's a cell wall, and then there's all this material. And inside, right inside the cell wall, there's a cell membrane. The cell membrane is mostly made up of fat or lipid. And then inside that, there's another layer of lipid on the nucleus, and the DNA is actually contained in that purple part called the nucleus. And so we need to get at that. All right, so that's what we're going to do now by extracting it. We're going to get the DNA out of the nucleus. All right, so the first thing we need to do is mash up these cell walls and mash them up. So I'm just going to take it right now, and I'm going to start by, in the plastic bag, what you want to do is mash up your strawberry.
Okay.
And don't pound on it or anything like that, because we don't want to bust the sac.
I'm making strawberry juice.
Right. So we're going to crunch it up, and It takes a little bit of time, but what we're doing right now is we're busting up the cell walls.
Okay.
And we're getting all the materials out of the cell walls, all right?
Okay.
And you could take the leaf off first to make it a little bit cleaner, but that's okay. It doesn't really matter. This will still work. What we want to do is have a pulp in here. So I've got this kind of liquid pulp.
Okay.
All right. So
kind of like a smoothie.
Kind of like we're gonna make a smoothie, except
slushy?
I'm about to make it not drinkable because I'm gonna pour this extraction fluid in there now. After I've mashed it up for a little while, I want to. I'm gonna pour some of this inside this bottle. It's mostly water, but I also have some liquid detergent in there.
Okay.
A little bit of that and some salt.
Okay.
And the reason why I have those things in there is that inside the cell wall, there's also this cell membrane. And it's also inside the nuclear membrane is also a membrane. And the membranes are made up mainly of lipids, which are fats. And what we want to do is get those out. And so liquid detergent is able to dissolve in water, and it's also able to dissolve in fat.
Okay.
And so your laundry detergent can take out stains, because you know how some things...
It attaches itself to the dirt, right? That's
Yeah.
what I've always said,
And
and then it moves it.
Yeah, so some things can dissolve well in water and some things don't, right? And so fats don't dissolve very well in water.
Okay.
But detergent will dissolve both of them. And so you can, if you put detergent in something or dish soap in something, then they'll dissolve, the fats will dissolve in it and the water will dissolve it as well. So you can get both of them to kind of dissolve in it and go away. So I'm going to put a small amount in here. Technically, if I was measuring something, I'd say, we're going to put 10 milliliters in here. But we're just going to try to get close here.
So if it attaches to, or it dissolves the lipids?
Dissolves the lipids, and so
Okay.
I'm gonna get the lipids.
So what do you want to be left behind after the lipids are dissolved?
The
is
DNA.
that the DNA is left behind?
Right, will be after I take it
Okay.
away. I'm gonna get it out of there. And the reason why there's a little bit of salt in there is because if I just use pure distilled water, then it's not the same concentration as what's in the cell. I'm gonna make it equal as the cell. So it's a little bit of dissolved salt
And
as
salt's
well.
a mineral, right? Don't
Yes,
laugh
and
at
it
me.
makes it so.
Okay. Okay.
It's what's called isotonic, which is the same concentration as the.
Do you want me to put some of
Yeah,
that in
let's
mine
put some in
too?
there, and then again,
At first I thought this was bubble water. Very
it
similar.
would make
except you
bubble
have
water. And what we want to do is we want to again close it up and mix it together a little
close it
more.
up. Is this something a student could do at home?
Yes, they could.
Okay.
They want to have parent supervision,
Mm-hmm.
and because we'll be using what's called isopropyl alcohol in a little bit. So far, we don't have anything that's toxic in here.
Okay.
I wouldn't want to.
I wouldn't want to
your
drink
bubble
this
soap.
soapy
I mean, it's like drinking soap
strawberry.
so
yeah.
far. All right, so you just mix that together a little bit, and that way we'll get the DNA separated from the fats in there.
Okay.
And now I have 2 cups, and we're going to make a little filter out of paper towels, and then you can just pretty much, wrap this around your finger,
Okay.
and then make a little kind of a cup that sits over the top of this,
Okay.
and make a little bowl, and then all you have to do now, and make sure you don't put a hole in it I thought I did. I didn't. I guess I just kind of.
Are we going to strain it? Is that what we're working
Yeah,
on?
we're going to pour it through here like
Okay.
your.
Could I have a paper towel, please?
You can have a paper towel too.
Thank you.
Here you go. And then go ahead and put that over your cup. And then what we're going to do is just pour this into the paper towel.
Okay.
Careful not to put too many bubbles in there, but
Not too many
yeah,
bubbles.
try to make it too bubbly is all. So I can pour it all in there and let it drip through. And it's going to take a little bit of time to drip through.
When I pour, do the bubbles stay behind?
I just don't want to make it too sudsy is all.
Okay.
That's all.
So don't shake it up a lot.
And don't and try not to squeeze it too much. I'm getting a pretty good amount. in
Does
there.
it? Oh, I put my finger in there. Does
That's
that
okay.
Little extra.
I know you're going to mix your DNA with the
Germs.
strawberry DNA, and they'll be able to
Then
trace
it's a crime scene.
it back to you.
Okay. So similar to that.
That looks good.
It smells like strawberry soap.
Yeah, it is. Pretty much strawberry soap.
Good point.
Okay, sometimes this takes several minutes to do.
Okay.
But again, what we're doing is we're straining out the liquid portion of this and
Can I
getting
lift
it
it
out of there. Yeah, you can lift it.
and see how it's straining it?
Yeah, it should be just dripping through. Mainly we're just trying to separate all that pulp from the liquid.
Okay. I've done this before when I made like alternative milk, like coconut milk. And
Okay.
you strain.
Oh yeah, you mix it together, you get pulp behind. Okay.
And you get the liquid out. Okay.
All right. Once you have a fair amount of it,
Use a
and
cheesecloth.
I mean, you want to wait for a good. I mean, if you're doing this at home, the longer you wait, the better it is. But
The
I think
longer you wait for it to strain?
yeah, because the more material you get, the better. But we're doing okay.
Okay.
And I can take this bag.
Should I squeeze it or not?
I did it a little bit.
Tiny bit.
Just as long as we don't want to break we don't want to break it and get it all the way through and get too many suds in it.
That's where maybe cheesecloth would be. Oh no. I thought you didn't want the pulp in there, but you don't want suds in there.
Yeah,
Okay.
I don't want suds in there either. Okay.
Good?
That's good. All right.
Okay.
So look what I've got. I've got, maybe, what?
1/4 inch?
Maybe about 15, 20 milliliters of it. That's fine.
Okay.
And so now I'm going to put it into a test tube. And of course, you don't have to use the test tube if you don't have one. I'll give you a test tube as well.
What would they use at home if they didn't have a test tube?
Like a small glass would work fine.
Okay, like a small.
Yeah. And then pour it in
see-through
carefully without
plastic?
trying to make suds. I'm
Okay,
trying to
so
pour
you tilted it so that it's.
I'm not going to try not to make too many bubbles. All right, there we go.
Is that a technique to tilt it and then it goes
Yeah.
down the side rather than
Yeah, so you don't make,
down the middle?
Yeah.
Okay.
it's like if you try to pour a soda without making too much fizz.
Yeah.
The same thing.
That's
All
where
right.
sometimes I put like a spoon into a bowl when I'm pouring something in there, and then it goes down the side of the spoon rather than splattering
That's right.
like something tomato. Okay, so this is our.
And you got a lot. That's great. You did much better meat. So now we've got our liquid in here, and hopefully it has DNA mixed in it with other things. And what will happen now is I'm going to mix it with some isopropyl alcohol.
Is that the same as rubbing alcohol?
It's the same as rubbing
Okay.
alcohol. So this is what you find in your medicine cabinet.
Okay.
And what this will do is this will cause, when it mixes with the DNA, it will cause the DNA to come out as a solid.
Okay.
It's not able to dissolve it. And what I want to do is I don't want to actually mix it with this. I'm going to just pour it down the side. I'm going to take about 3 or 4 dropperfuls.
So you
of
have a
this.
nice dropper to get.
And I'm just going to let it pour down the side and then form a layer. So I should have a clear layer on here. And then a
clear layer on top.
clear layer on top, and then a pink layer on bottom. I'm going to put actually one more in there.
So what's the isopropyl alcohol doing?
It's getting the DNA to come out as a solid, so it doesn't dissolve in there. It really helps if this is super cold.
Is it bringing it to the top?
It will mix. Yeah, it will mix. It'll mix with the stuff that's there too, and cause some of it to come out.
Okay.
And it's helpful if this is really, really cold, and I wasn't able to super chill it, so.
If it was cold, what would make the difference?
I think it just helps make it solidify better.
Okay. And then the solid particles that are going to form, that's where the DNA is? Or is that the chromosomes?
It's where the
DNA?
actually the DNA is going to be, it's not going to be in chromatin, so we're just going to have straight DNA if it works.
That's amazing.
We'll see though. We'll see if it's amazing or
We'll
not.
see if it works.
All right, so what we should have in here, and this takes some time sometimes, but we should have in here is we're going to see this clear layer. Oh, yeah, I've got some. I can see it already. So
those are
if we have
really
I don't know if you can see the close-up of it, but if you see,
Let's see how it'll look on.
if you can see
Something black would be better.
I don't know.
Let me try the back of my.
Yeah, you can see it. But if you can see we have a layer here. We have more the pulpy
go
layer there, and then we have a
forward.
clear layer on top of it. And you can see in that clear layer, it's starting to come out.
It's kind of foggy, and that's why.
You see a little bit of fogginess
So
to
that's the DNA. Oh
That's
my
the DNA, and we'll be
goodness.
able to pull it out here in a second.
With like a tweezers or what?
With my skewer.
A skewer.
Yeah,
and
we're gonna be able to get it
it'll
out. You can see that little layer. You see that little thin layer of almost looks like bubbles right there. That is DNA.
And I don't want to mix it, right? Because it'll get bubbly.
Yeah, if you stir it around a bit, you're going to lose it.
Okay.
So,
Okay. Wow.
It's pretty cool. So let's see what we can do here. I'll give you one for yours.
Okay.
And then I'll take one here, and let's see how we can do on this. But you should be able to pull out just some of that goop. I've got some right here. You can see that.
Goop's hard to see.
I've got a nice little blob of it.
Do you? Oh, it's like slimy goop.
It's like slime.
Tell that to my daughter. She'll want to make slime out of it. Can they see it in front of the black?
Then I got to show the black.
Oh yeah, it's like slime. I was going to say something else, but
right. Well, yeah. If you blew your nose.
It's like a teardrop dripping down.
A teardrop dripping down. Okay.
Yes.
This is DNA. And so if you get a little package and stuff, this is strawberry DNA that's sitting there. Now, if we waited longer, we'd get more of it forming out. If we had it colder, we'd have a little better bit. But this definitely is.
You didn't drop any DNA on the keyboard,
did not drop any DNA on the keyboard.
This is really neat.
Cool?
I'm going to try to grab some of mine.
Yeah, let's see if I can get any more. I mean, you can get, and I just twisted a little bit. Just to wrap it all up.
Oh, I'm trying not to. So you said twist it instead?
That seems to help. If I twist it around a little bit, I get some more.
You have it tilted.
Yeah, I just have a little bit at the end of it is all.
So it's not the chromosome, right?
It's not. It's just straight DNA. And of course, DNA is this big, very long chain molecule.
Nope, I don't got much.
Okay.
Or I don't have very much.
It's. I mean, it's pretty well. I mean, if it's colder, it works a lot better. I just didn't get a chance to cool it, but there's some on there, and we got a good chance to see some of it. Yeah. So there you go. This is how you can extract DNA from strawberries. And again, it's sitting inside that nucleus, and so you got to be able to like tear away everything else.
Separate everything. So the lipids went away.
The lipids went away, and
the nucleus of the cell went away.
And all the cell wall material went away.
The membranes
all
went
the fiber,
away.
which is made out of fiber, right? And so we got rid of all that.
And they're not gone. They're just down in here in the soapy.
Some of it
mixture.
is, yeah.
Some of it, okay.
Yeah. So, and again, of course, we recognize DNA to look like this, but really, it's quite clear when we see it. And so the question is, how did they figure out that it was like this? And they did a couple things. If you look at this model of DNA, you'll see, first of all, it's this thing called a double helix. It's twisted around itself. There's two sides of it. In the clear band side, it kind of all looks the same, and then they have these little spurs coming off of it.
It kind of blends in with your shirt.
That are both yellow, red, blue, and green. Of course, that isn't the way it would look. But when they're trying to figure out the structure of it, they had a couple clues about it. First of all, I don't know if we can look at get the whiteboard going here.
The next, okay.
That if we had a couple of things about DNA. So they knew what DNA was made out of. They knew that it had three parts to it. It had a. You want to write? Oh, sure, you can write.
Okay.
It had a 5-carbon sugar. So it had a sugar. It's made out of a sugar. This is where the deoxyribo comes in. DNA is made out of deoxyribose.
Okay.
Ribose is like sucrose is a sugar, and glucose, and deoxyribose is a sugar. That's the D. The second thing they have in it, it's called a phosphate group, that letter P, the phosphate group. And then the third thing they have in it is a nitrogenous base. So this is each part of the DNA.
I may need your help.
So just you could say a base.
Just a base.
You can say a base, and it's a nitrogenous base, which means it has nitrogen in it.
Okay.
And so they all need all pieces of the DNA, each section of the DNA.
Capital N would be nitrogen?
Yeah.
Okay.
Each section of the DNA had these three things in it. And so usually it's depicted that the sugar and the phosphate group are along this plastic thing, and the nitrogenous base is listed as one of these blue things in the middle.
Oh, thank
and so what they found, there's 4 different kinds of nitrogenous bases. And there's adenine, guanine, cytosine, and thymine. And so usually if you put down letters, it would be G, C, T, and A.
G, C, T, A.
And A.
Okay.
And so one of the things the people who are trying to figure out the structure of this, one of the big clues they got, and this is where the math comes in, so I thought you'd like this.
I would love to hear the math.
A little math clue is this. This researcher got interested in studying DNA and what made the structure of it, and his name was Erwin Chargaff, and he made what's called Chargaff's rules.
I would love to see how his last name's spelled sometimes.
Chargaff, C-H-A-R-G-A-F-F, I believe. G-O-F-F, C-H-A-R-G-O-F-F. And this is what he found. If we could go write these, if we can make a little table.
We have a little bit of time.
We have a little bit of time, yeah.
Okay, so a table, and what's on the table?
And then we'll just say, just put down 4 columns.
4 columns, okay.
And then put down A.
So A.
T.
T.
G, C.
G, C.
Okay. And then the first thing, let's look at humans. And so this is the composition of humans.
Okay.
And it was, on humans, it was 31% A.
Okay.
Pretty much, I'm going to get around up to whole numbers. 29% T. 20% G. And 20% C.
Okay.
This is for humans.
Okay, I think we have time for one more.
And then let's put down, how about a turtle?
Okay, the turtle?
Was 30% A, 28% T, 22% G,
22.
and 21% C.
So a little bit different of the nitrogenuses, or the nitrogen bases.
But did you notice some things? Do you notice some things about it? But for every living thing, it kind of had the same kind of ratios.
Yeah.
So close to the same amount of A and T, and close to the same amount of G and C, guanine and cytosine.
Mm-hmm.
And so from that, those proportions, and you looked on every,
So
you looked at all.
These two are close, and then these two are close to each other.
And so from that, the people that discovered how the structure was realized that this pairing must have been happening. That G and C were
Oh, and so that's
combined,
where the colors.
and then the colors are the same, and T and A were the same.
So green and blue all the way along here, red and white. I mean, not red and white. Red and yellow.
Red and yellow were always paired.
Mm-hmm.
And so that was a big clue into that.
And those are the bases.
These are the bases were paired together and actually crossed together, and were able to form this double helix structure because of that. And there's a bunch of other things that went into it.
So if you want to know more about DNA, we can look at the book.
And then
right?
there's lots of other things.
Lots of studies. DNA is, everything's made up of DNA.
Everything, all life has DNA as part of it. And it's a fascinating molecule. How it divides, how it makes proteins, how it works is, they're still learning more and more about it now.
How exciting.
All right, so we'll see you on Thursday. Until then, have a great rest of your week.
Yay.
And bye.
Thanks for watching.